grow, what was it? Grow fungi everywhere, every place around the planet. Grow fungi every place around the planet. So here we're with, write them all down one time. Rhizofulvin, flucocytosine.
Sure, yeah, thanks.
The more you write and erase it, and write and erase it, and write and erase it, the better off you'll be. Do you guys want one of these, or you guys have your own? I'm sorry, what? Oh, you're good. Okay, cool. Rhizofulvin. So we're not going to talk about these again today, so that's why it's kind of beneficial. I mean, you can do this on your own, but it does help to just go through them together one more time. Rhizofulvin, flucocytosine, echinocandins. Sometimes those are called fungins. But you don't know that. Just echinocandins is fine. Polyenes. For polyenes, do remember amphotericin. I want you to remember that because you'll see that over and over. You'll see amphotericin is like one of the primary antifungals that are used. Azoles, so I do want you to remember that azole because that is all topical antifungals for any kind of cream or anything like that. Those are generally azoles. Terbinafine and polyoxidants.
We don't need to know any like examples or anything for these books.
No.
But we do need to learn the mode of action.
Yes. So if it helps you to write the names one more time, however you want to do it. So this would be a lot for me to memorize in one go. For me, I think I'd probably take it apart like 2, 3 at a time and try and memorize 2 or 3.
So if it's not like broad or narrow spectrum, would you present this like in in an exam? Would you say I want to treat it with this mode of action? Which antifungal would I use, or how would you present that?
Yeah, so it could be with this mode of action, what antifungal would you use, or this antifungal, what is its mode of action? Or as we get to the final, it's like this infection, is this a bacterial or fungal infection, and what would you treat it, and what is the method of action? So then you'd have to know, is that a bacterial or fungal infection?
Yeah.
What is the prescription that you would prescribe?
And if it's like a topical fungal, then you would probably use this whole range.
Yeah, but you don't have to know that. You just,
Okay.
I would make it either name one randomly, or if I said this is the mode of action, know this specific antifungal or something like that.
Okay.
Yeah. Okay, so rhizofulvins, microtubules. So however you need to do like probably what I would do is write it. So I would do grow fungi every place around the planet, and I would write them over and 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 over until I felt really comfortable with that. And then when I moved on to mode of action or method of action, I would go M D C E E E C. So I'd go M D C, or it says cell wall glucans. I'd probably just write glucans. And then DNA replication, I would probably do an R. Replication, microtubules, replication, glucans, ergosterol, chitin. Is probably, I would remember like, yes. And then when I was right, rhizofulvin, flucocytosine, I'd write that so many times that I was really comfortable with the order and how to write those. And then when I went to the mode of action, this is how I'd give myself a little chart. So then I'd know rhizofulvins, microtubules, flucocytosines, replication, echinocandins, glucans, polyenes, ergosterol, azoles, ergosterol, terbinafine, ergosterol, polyoxidants, chitin. Is probably how I would do it. do it how you would memorize the best. Yeah, go ahead.
For the micro pad on your chart,
That's for finals,
oh, it's for the final. Okay.
Yes, yes.
Can you explain the highlighting?
Explain
You have
the
like a light
highlighting.
highlighter and a
Oh,
dark
yes, so
highlighter?
that you don't have to know. So the highlighting, just like on the,
Yeah, that slide.
yeah, so just like on the backs of the note cards, you don't have to know any of that.
Any of it, okay.
You know, you will have to know like diagnostics, like it's probably, if it's fungal, it's probably skin, it's probably pneumonia, something like that. But you don't have to know like, candidiasis you do because that's our note card, but like cryptococcus, it causes this, this is the organism. Mycotoxicosis, it causes this is the organism. You don't have to know any of that, just know this. So I'll do it on the final the same
Wait,
way.
what do you want us to know?
Similarly, I'm sorry, what?
I'm confused about what it is we need to know.
So just the name itself. So what I'll ask is similar to the final, how I did it on the midterm, is like identify what is a fungal infection, what is a viral infection, what's a bacterial infection.
Okay, so like, if you're asking the question, you would say the patient has mycotoxicosis?
And you'd say.
Or you would say-
That is as a result of a fungal infection.
Okay, so you wouldn't do the things that are in parentheses.
Yes, nothing in parentheses, yeah. So I wouldn't say your patient has pneumonia, what could it be? And you have to say cryptotoxicosis. But if I said pneumonia, you'd say, that's probably fungal. Because we know fungal organisms are often pneumonia, but also bacterial. It would be bacterial, viral, fungal, and then as we get later on, the other organisms. So
But if you said pneumonia, how would we know that that was fungal if all of them can cause it?
it would be like I did on the midterm, where it's like multiple. It's not just one can cause that.
Oh,
Do you
okay,
remember
so
how
we
did
would
that?
say all of them could cause it.
All of them can cause it.
Okay.
Yeah. So it's just a good idea to just kind of read through, the tinnias, but that's our note card, so you'll have to know those for the note card. But basically, they're skin or pneumonia. Yeah. So, if it says your patient has cryptococcicosis, then you'll say it's fungal. Where that might trick some people who did not memorize that cryptococcicosis belonged in mycopad, because it kind of sounds like bacteria, you know, that kind of thing.
Sorry, are these, fungi?
They're all fungi.
Okay, 'cause
I thought they were fungal. Aren't they fungal illnesses?
So they, the way that they
This is
name.
condition.
Yes, so they're conditions, that's how fungi infections are named.
Okay.
So it'd be like if you had Staphylococcus aureus and they'd call it Staphylococcus aureus.
I knew I was
You know,
gonna
like that's how they
Yeah.
name fungal infections. They name it after the bacteria, or after the fungi.
How are you
Okay,
today?
cause
So-
like I was looking at it,
Did you
like,
think that you
these
were?
look like conditions.
Oh, yes.
And I
Oh,
was
I
looking
brought-
for like kind
if
of
you
talking
wanna
about them, like
take a
they
look
were
at them, yeah.
like fungal like infections or something. I was like
Taking
super
with you.
confused, but I asked this question,
Oh, yeah. So here is my little disclaimer, but it's not very clear. I know this was gonna confuse people, that sometimes they're named after the fungi itself. So, but sometimes they're not, which makes it just confusing. If you don't wanna stress about that,
It's all- it's literally
just
all-
memorize that. Just memorize these are all fungal infections, that's what they
But
are.
that would
Yeah.
be, that helps narrow it down, especially for the cryptococcicosis.
Uh-huh.
Because it's a condition which means it's fungal.
Yes.
Okay.
Yeah. And then myco always kind of tells you that it's fungal, but they're not all myco either. A lot of them are called myco. So if you see that word on a quiz, then you're like, or on a final, then you're like, oh, myco, that means it's fungal for sure. But not all of them are myco.
The P is mokai.
I'm sorry, what?
The P says mokai.
Well, right now we have like half the class getting A's. I would imagine most people do well. The finals, it's harder. It's harder because a lot of people wait to study these. And diagnostics can be tricky So the reason why diagnostics, I always try and bring this up as we get to the end, because this is what, and so I tried to incorporate this a little bit more in the midterm, so that maybe when you guys read over the midterm again before the final, it'll help you understand how I'm gonna ask diagnostic questions. So students have a hard time when you're like, okay, on the front of your note card, it says Staph aureus, and on the back of your note card, it says skin, it says pneumonia, lungs, it says gastrointestinal. So what students have a hard time doing is flipping the note card the other way around. When I say, is it a skin infection? Well, what can it be? Because they remember the front. For some reason, it's just a mind shift. Like, I don't know how to help with that, like how to help prepare you then for that. Do it both ways.
Yeah,
So then you're like, if it's.
Memorize it by the bacteria and what it does, and read the cards backwards. It would be like by isolation.
And then ideally, that would be a great recommendation, but ideally also making another note card that says skin and all the organisms that it could be. So making another set of note cards that said skin, and then it's like, we didn't use epidermidis as a card, but like, you know, anything that's gastrointestinal, write down all of our gastrointestinals that from our note cards. Like anything that's a sexually transmitted infection, like STIs, writing down the STIs, because we have a few STIs. So going the other way around is important.
So diagnostics is a big part of the.
Yes, diagnostics is a big part of the final. The infections, like the bacteria look like plums, virus, McCaffey. I can't say without with a straight face because it's so dumb. What else was it? The mycopad, and then protozoa. I can't remember what the protozoa was.
Yeah, so not only diagnosing like what it is, virus, but like the actual bacteria virus.
Yes, if it's part of our note cards. Yes, yes. So if it's like cryptococcicosis, that wasn't one of our note cards. But the reason you have to know it is because you have to know it belongs in the fungi. Yes. But you don't have to know any details about it. All you have to know is it's a fungus.
Oh, yeah.
Yeah.
Okay, and we're like halfway
Let's see how many more we have left. We just did dermatophytes, right? So coccidioides we are doing actually, which I forgot I put on the next. No, I might keep it. Let me see how many more we have. So, 1, 2, 3, 4, 5, 6, 7 left.
Are all of those in the file that you posted? Even the ones?
I just posted today everything.
Okay, cool. Thanks.
So why, oh, you know why I brought in coccidioides is because it's going around right now. That is why I changed, I added this. Okay, so let's get started. I don't know where I'm missing. So what I would recommend doing is making yourself some note cards or whatever you want to do to prepare, and just start going through them once a day. Even if you go through your note cards once a day and you don't really focus on any one note card, or you're like, oh gosh, I'm so far from memorizing that. Every single time that you touch on that information, it will help you. It will beyond measure help you, even if you're just doing it while you're in line at the grocery store. Okay. So, I'm gonna finish this today too. So what I I started to do, I didn't finish here with fungi, but I started to kind of write everything that I know about bacteria. So it might be a good idea for you just to reorganize your thoughts a little bit, and maybe even just do like one whole front and back page bullet pointed of like things that you need to know or remind yourself of about bacteria. And then I wrote as much as I can think about having to do with viruses. So let's just do a quick bullet, and I'm gonna do fungi really quick. Bacteria, they're prokaryotes. So what does a prokaryote mean versus a eukaryote? Cell walls are made of peptidoglycan. That's what's unique about bacteria. Gram-positive, half thick. Gram-negative, half thin. Gram-negative have an outer membrane. It makes it difficult for them to be treated with antibiotics. They can have a capsule, a slime layer, a biofilm made of that sticky glycocalyx. So these are like my notes as a student. They would just be my little cues of like, oh yeah, don't forget to study that. Plasmids, we're gonna go over plasmids to death today. Antibiotics, antibiotic resistance, review those, review that. Aerobic, anaerobic, facultative, review our metabolism. You guys are so smart. Look how much you guys have learned so far. I love it. I love it. Okay, so cell structure, nucleoid, 70S ribosomes, flagella, pilus, fimbriae. Those are just ones that I can think of off the top of my head. Replication, they have one origin of replication. DNA polymerase 3 is the one. There's 4 types of mutations, I think. Is there 4? Yeah, that's right. And then the other enzymes, topoisomerase, single-stranded binding protein. Transcription, they're formed into operons. Operons, remember the pieces of the operon, remember the lac operon, the trip operon. RNA polymerase makes RNA. Don't forget RNA to DNA, how to write that double-stranded DNA to an RNA. Translation happens with 70S ribosomes, forms amino acid structures, forms proteins. Transmission, so that might be a good thing to go back over. What is a biological vector versus a mechanical vector, that kind of thing. Cytopathic effects, so our exotoxins and endotoxins. Bacteria look like plums, so it's a good idea to start making note cards on that. What are bacterial infections? And then our diagnostics, which we didn't really cover diagnostics of bacteria until we got to viruses so that we could compare the two. Then we talked about viruses. So viruses can be naked or enveloped. Naked, they can be helical or polyhedral capsids around the DNA or RNA. Enveloped, they have these inside the envelope. That's what this little arrow means. There's a phospholipid bilayer on the outside and spike proteins. Enzymes on the inside, they can have replicase, they can have reverse transcriptase. Those are a couple enzymes they come with. Remember what those do. Baltimore classifications, just knowing what that means, DNA versus RNA, and some of those questions were how one type of classification might get to the RNA so that our own ribosomes will replicate it. So just some of those questions I like to ask because it makes you really think about replication, transcription, translation. DNA versus RNA mutations. So RNA are more likely to mutate because they don't have the DNA proofreader. Zoonotic diseases, antigenic shift, drift, and what that means. Lytic versus lysogenic cycles of viruses that they can incorporate into the genome, or they can break out of the genome and start replicating. Cytopathic effects of viruses were the proviruses, oncoviruses, syncytia. And then vaccines, I think there were 5 that we memorized, I think so. Antivirals were the VIRs. DDX, so that's when we looked at, is it pneumonial, is it bacterial versus viral pneumonia? We can really tell what is otitis media, what is a respiratory infection look like, what is, so just some of those diagnostics comparing bacteria and viruses. Transmission, so how your patient might have come across a viral infection, so reviewing transmission, they're similar. And then virus McHappy were all our viral infections, if that helps you memorize that. Okay, so fungal, we have yeasts and we have molds. I wanted to put here, they're single cell cell, or they have pseudohyphae. These are facultative. They are, anything else we need to know about those? They can sexually or asexual reproduce. Let me get a better green. Okay, so molds, we have hyphae, mycelium, they are aerobic. I wanted to do this before I forgot. Manoproteins, chitin, and glucans, and ergosterol. A lot of times they are pneumonia or skin because they're aerobic. They can also be like vaginal, maybe. I'm not really sure what to go through here because I wasn't looking at my cheat sheet. Pseudohyphae, facultative, sexually asexual. So what I would do to complete this, I can do this next time too. I can write it out one more time to help you. But where I was getting all this from is the study guide outline. So just helping kind of associate that. Oh yeah, maybe just let me look at that 2 seconds. Just let's finish it. I don't like when things are unfinished. It bugs
Oh wait,
me.
here's more.
Is this it? Oh yeah, fungi. This is all I need.
We got more.
This one, oh yeah, that's diagnostics. We don't need that one. Just this one. Okay, so dimorphic was temperature. Microscopy was spaghetti and meatballs. And the meatballs are the fruiting bodies, so those were the sporangiospores, conidiospores, CPE were the mycotoxins, enzymes, and direct infection. Here we first talked about meiosis because we talked about sexual reproduction. We talked about the problem with fungi was that they are airborne and they're commensal, they're everywhere. So then know your antifungals, which we went through together. I'll show you the thing of it. Antifungal resistance. Anything else? Mycopad. Okay,
What
so
about
that's.
horizontal gene transfer for bacteria?
Yes, that is important too. Let me add that up there. We'll talk about that a ton today, actually. That's treatment, I should. Okay. Okay, so that's maybe a good idea to start now before the final. Kind of organize yourself. Make sure that you know, like, you've very, very clear. That's what I really think is important getting out of this class is what is each thing. You know, if you walk out of the class and you don't really know what a virus is, I haven't done my job. So making sure that like each category is, that you're clear on the categories and how those are differentiated. Okay, so we are sure you're very clear on fungi. We're going to bring that back up again for the quiz. So the quiz is going to be half fungi, half protozoa. So I'll put on the study guide outline for Friday what topics that we hit so that you know what to cover. So protozoa are going to be on the quiz. So try to be with me as much as you can, get some visual memorization going, right? However you need to do that, because we have, this is what we practiced. Antibiotics one more time just for funsies, and then antifungals, getting to memorize those antifungals and the method of action. The method of action we did, this one was microtubules, this one was,
RNA.
oh yeah,
sorry, DNA.
replication. This one was
No, it's cell walls.
cell wall glucans,
And then E, and C.
ergosterol,
And then chin.
or chite. Okay, so the reason I wrote that out is because this is how I would memorize it myself.
I think
I
this
guess.
is.
I think there's 3 E's.
Oh,
Yeah.
3 E's.
Yeah. Ergosterol and then.
Ergosterol. Okay. Okay, so that's how I would memorize it myself. So practice that. Okay, awesome. So moving on to protozoa of a genome. So here you can see there's an origin of replication. What binds here in a bacteria? Sorry, say that again. Oh yes, that yeah, that's the Tata box. That it is full of A's and T's. But what will bind here? Yeah. Awesome. DNA polymerase 3 will bind here. Also the helicase, topoisomerase. That's what this origin is for. So we have to put in an origin of replication, right? If you have one plasmid in one bacteria, that's not going to help us. The bacteria has to replicate the plasmid genome just like its own genome, right? So we want to trick the polymerase into thinking this is its normal genome. Yeah.
So this is it's going in both directions, right?
Mm-hmm.
So is it also like flipped, like one for the 3' side and one for? Does that make sense? Like, because the way you're looking at it, it just looks like it's just like one.
Oh
Like-
yeah. Yes, I think that's always really important to bring up because we look at things in cartoons and two-dimensionally so much. You know, we look at like what is a cartoon of it, but you can't look at the real thing. So it's very difficult to understand that. But yes, this whole plasmid is a double-stranded DNA. So it's always double-stranded unless you have some weird virus where it's single-stranded. So then when you have your origin of replication, and now we have this bubble, replication bubble, and now it will go bidirectionally in both directions, and now then you'll have one parent strand and a daughter strand, and you'll have a parent strand and a daughter strand. So remember, it's semi-conservative, which means it's going to conserve one strip and make another copy to match it. Conserve one strip and make another copy to match it. So now it's double-stranded DNA. Very good question. Okay, yeah, so it's important to remember what you were actually looking at. Always remind me to bring that back in because that can be very confusing. Okay, so here we started with the origin of replication. Here's the promoter. So this promoter, what it is what binds to a promoter? What's it? Wait, say it. No, not the repressor.
Is it the RNA polymerase?
RNA polymerase. So you have to make an RNA polymerase binding site. Why isn't there a regulator or an operator? So remember RPO. Why isn't there an operator or a regulator?
Because it's a continuous cycle.
Yeah, because why do we want to stop it? And sometimes people do stop it. Sometimes you would want to. Like when you're studying, for example, development of human beings, like development in utero, then you do it in a mouse and you would stop transcription or translation at some point so you can see what effect that protein had on a developing fetus versus an adult. So then if you stop it at one point and put in a repressor, then you're like, oh, well, that was fatal to my mouse, it must be important in development at some stage, so you can study it that way too. But for the most part, we don't include them. We just want it to go and go and go and make as many protein products as we can. Here's the restriction site. Restriction sites are where restriction endonuclease cut. So the restriction endonuclease will cut at restriction sites. So we have to put in the site that matches the eco R1. So remember, if we have a bacterium, we have one, and they have this enzyme, then what we're going to do on the plasmid is we're going to make this site an eco R1 site, which is just a series of A's, T's, G's, and C's, so that we can cut this section out and put in our gene of choice. So we use the restriction endonuclease of a bacteria, we cut out this section according to these sites, and we're going to put in our gene of choice into that little section. So, what else do I want to say about that? Yeah, I guess that's it. Let me know if you have any questions about that. And then we're going to add what's called a selectable marker. So we'll talk about what that means and why. So plasmids, you can just buy plasmids from a plasmid store, plasmid.com. You can buy plasmids, which is cool because we can buy them. We use them in the science all the time and to do whatever you want. But it's also a little bit terrifying. You know, it's a little bit like, you know, how much access, this is what I love about the documentary, how much access are we giving to the public, and why would it be bad to give access to the public, and why would it be good to give access to the public? So should I spoiler alert for you of why? Okay, so in the documentary, because some of you probably won't have time to watch it, it's kind of long, but what they're saying is, okay, you go through this whole story of this guy who's doing genetic engineering in his garage. So he's doing genetic engineering on dogs, and he's breeding dogs and trying to make glow-in-the-dark dogs and like genetically modifying them for different purposes, and like how are you going to manipulate this dog to have all these crazy genes to do other things? Who knows what he's trying to do? But so he's in his lab, and you kind of get this feeling of like, oh my gosh, this is scary, you know, that he can make his own plasmids, or he can buy them online, and he can genetically manipulate anything he wants. People, like, I mean, it's against the law, but it's not against the law to work on dogs, and it's not against the law to work on bacteria. It's not against the law to work on viruses. So, you know, what is in the hands of the layperson, of the actual people who are doing this work? So that's, you start off like terrified, like, oh my gosh, this is so scary. He's going to make some sort of genetically modified dog or virus or something. And then it goes into this whole story of a child who, I think it's been like 2 years since I watched it, but I think he's got vision, he's blind. And so he went through his, it's this genetic, they did some genetic engineering on this protein that would help him basically see again. And I think he's like 3 or 4 or something like that, and his parents are like, why can't we get the funding for this? Why is this not more readily available? Well, it's only because there's only like 10% of the population that has it. It's like 10% is huge, like 1% of the population. So these families are like, well, why can't you manufacture it? They're like, well, it'll cost us millions and billions of dollars, and it's not financially worth it for us to do it. And they're like, you know, you get like your total heart into this kid, and you're like, well, why is that fair? Like, why can't this guy in his garage make the gene that will help this kid? And like, and then it goes into this whole other story about how this family just went broke, spending millions, like tried to get all this funding for these pharmaceutical companies that are making billions and billions of dollars. And so it's this whole cycle of how crazy genetic engineering is right now. But basically being said is that you can do genetic engineering yourself. We are gonna do it today, and yeah, you can form your own opinion about whether you think it's cool or not. Yeah.
So the Eco R1
Mhm.
is an enzyme.
The Eco R1 is a spot on the genome. So here,
So
let me show you.
that's the restriction site.
Yeah, so here's the little restriction site. This is CTAG, GTAC that it will cut, and this CTAG and GTAC. So these are Eco R1 sites where the restriction endonuclease will cut.
Okay, so the enzyme is the restriction nuclease.
Yeah,
And it recognizes that
and cuts it.
Okay.
Yeah. Okay, so we'll do, if we have time, we'll do a little review at the end, but also we